Hi everyone and welcome back to another video. My name is Jane and I'm a second year PhD candidate in cancer research. Good morning everyone and welcome back to another video. Today is Monday the 23rd of September, I do believe. I have just arrived at the lab. So the first thing I will do today is pop into the lab and check my cells, make sure they are all growing healthily and happily. So today I'm planning on setting up an experiment called a colony assay. And this is an experiment where you basically take a really small amount of cells. I'll probably do about 3,000 cells. And you put the cells onto a plate, and I will hopefully be able to show you the plate. And then you let them grow for 2 weeks, and the cells will undergo different conditions, and then we will compare the conditions to see if there is a different number of colonies that form. So if the cells are happy, then the individual little cells in the plate should grow into little colonies, and then hopefully in 2 weeks we'll have some good results from that. It's sort of a bit of a preliminary experiment. I'm just testing to see if I can get it to work, because the cell line I'm working with are notoriously difficult to do a colony assay with, so we'll see how that goes. I'm also going to be making some gels so that I can run some protein on the gels this week and do some protein analysis. I'll probably make up my protein samples today if I get time. I have a lot of samples to make up, I think 12, which doesn't sound like a lot, but each of them needs to be pipetted at different volumes, so it takes a bit of work and a lot of labeling of tubes. I will also harvest some cells to test them for mycoplasma. So I've been growing cells without antibiotics for about a week now. And so mycoplasma is basically an infection that cells growing in culture can catch, and it makes them sick, and it also makes them unpublishable. So I will be checking my cells for mycoplasma, or submitting them to check them for mycoplasma today as well. Yeah, I need to sit down and plan some upcoming experiments as well today. So fingers crossed I get to take you with me for some of that. Hi everyone. So today is Tuesday the 24th of September, and as you can see I am at home. Basically, when I got home from the lab yesterday at about 6:30 p.m, this cheeky little monkey, the one sitting at the back, so her name is Lucy and she is an epileptic dog. We think that Shani, sitting in the front, may also be epileptic, but her little tremors or seizures are not as severe as Lucy's. So Lucy has life-threatening seizures. She is currently being treated by a veterinary specialist, which is fine. She's okay. But so last night when I got home, she was having a little tremor. She was fine after a couple of minutes, but after she has a tremor, I prefer for someone to be with her for 24 hours, just in case she does have another seizure or another tremor. And so the other reason was, when I woke up this morning, I kind of felt a bit under the weather, and so just everything combined, I thought it would be better for me to work from home today. And one of the benefits of working in academia is that you can work from home sometimes if you need to. So I had done all the lab work that I really urgently needed to do yesterday, so I didn't have anything urgent to do today. So, cheeky girl. Yeah, so it was okay for me to be home today. I did some reading, I caught up on the literature, and I really like to do this when I'm at home because it's peaceful and it's quiet and there are no interruptions, and I can really focus on using my brain and thinking about what I'm reading, which is great. So yesterday things didn't exactly go according to plan. When I checked my cells, they looked a bit funky, and it's hard to describe how they looked funky, but I did take a picture of what they look like normally compared to what they looked like yesterday, and they just looked a bit off. So I basically took the cells out of the plastic flasks that they grow in, and I diluted them and I put them back in the flask, and I will check what they look like next time I'm in the lab. If they still look funky, I'll chuck them in the bin and bring up fresh cells from the freezer and make fresh cells if because I don't have all of those cell lines frozen, which is unfortunate because it will take me some time to make those cell lines again, but it's better than working with cells that I'm not confident with, so that's fine. And I submitted them for mycoplasma testing yesterday, which I mentioned as well. So if they're mycoplasma positive, I will chuck them out. So mycoplasma can make them look a bit funky as well. So we'll see how that goes. I froze down some cells, which I took a video of. I made some gels, which I mentioned I was going to do. I set up my colony assay, which I also mentioned. I'm not sure if this will need to be chucked out because those cells also looked a bit off. And I did not get to make up my protein samples because I ended up stuck doing cell culture for 3 hours. So I was exhausted by the end, and I didn't finish until 6 o'clock last night, so I was keen to get home. So that's about it from me today. If I'm feeling better tomorrow, I will go back to the lab tomorrow. If not, I might continue reading at home. We'll see how we go. Yeah, and that's all. See ya. Good morning, everyone. Today is Thursday, the 26th of September, and I am back in the lab. So it's 8:30 in the morning. I stayed home again yesterday because I was still feeling under the weather, and I pretty much just read some papers and did some, like, I guess PhD like admin work stuff, like filling out forms and boring stuff like that. But so today I'm gonna pop downstairs into the lab and check my cells. It's been a few days since I've seen them now, so I'm interested to see if they still look a bit funky. Today I'm planning on actually making up those protein samples that I talked about last time and actually running some gels today. So I'll need to run 4 gels and that will probably take me about 5 hours to complete the whole experiment, and I'm hoping to take some video of that today too. I guess wish me luck, and we'll see how we go. Hopefully we should have some good results tomorrow from these protein gels. So I checked my cells this morning and they definitely still look funky. And I'm pretty sure that one of my cell lines has infected one of my other cell lines, which is pretty embarrassing, and I've never had it happen before. But basically I'm going to chuck those cells out and bring up some fresh cells. Here are my fresh cells, still frozen in their little vials. So I'm going to defrost these, and then hopefully they're not contaminated. Good morning everyone! Today is Friday the 27th of September. Thank goodness it is Friday. So sorry about all of the background noise and the wind and things. There's trains going past. I got kicked out of the usual room that I film in because I think there is a meeting in there, which is fine, whatever. But usually on a Friday morning at 9 o'clock we would have lab meeting. But we don't have lab meeting this week because the boss is away. So today I'm going to pop into the lab and check the cells that I brought up out of the freezer yesterday and make sure that they're okay. Hopefully they haven't been contaminated by my other cell line, which would be great. So the colony assay that I set up was with the cells that I have now thrown out. So I might need to discard that experiment as well. I should have some results from the protein gel that I ran yesterday, so I'm pretty excited to see those results. I'm not sure what to expect, but I'm looking forward to seeing the results. And so I'm gonna have to sit down and plan some experiments. I haven't sat down and done any planning this week yet, but yeah. If I need to remake those cell lines, my other 3 cell lines that I didn't have frozen, I'll need to plan that, and there's a lot of steps involved with that, and I need to bring up like additional cell lines, like different cell lines. It's complicated. Anyway, it doesn't matter. Sorry for the bad lighting too, it's really bright out there. But the view is beautiful, I'll show you. So I appear to be the first person who has arrived in the lab, which is great because it means I can wander around and film without feeling like an idiot. So the first thing I'm going to do is get my protein membranes from the cold room. Let me show you what this looks like. So the first thing I'm going to do with these membranes is I will wash them 3 times in a washing buffer, or 4 times maybe, for 5 to 10 minutes each. And while they're washing, I'll put away all the equipment that I used yesterday and check my cells. and then I will put the secondary antibody on for an hour, and then I'll wash that 3 or 4 times, and then we should have some results in a few hours. It's results time! For some reason I'm alone in the lab again, so I'm filming again, and it's so nice and quiet. So I hope you enjoyed following me around in the lab this week. This is a pretty classic example of how things can go wrong in the lab and how we deal with the issues that we encounter. My mycoplasma results ended up coming back negative, which was some really good news, so my cells are not infected. My protein results were simultaneously good and interesting and expected and unexpected all at the same time, so I will need to repeat that experiment in the coming weeks. It's pretty common for protein results to be all kinds of interesting and unexpected. And so the cell lines that I brought up on Thursday were looking good on Friday, but I'll need to give them some more time to grow out before I will know if they are contaminated with another cell line. Fingers crossed that they are a pure cell line. So if you enjoyed this video, please give it a thumbs up, and if you would like to see more videos like this, please hit the subscribe button, and happy sciencing!